Welcome to Lieven Kempenaers' web seminar program. My name is Birgit Toscher and I will be your host for today. The title of today's webinar is Successful Elemental Analysis Using XRF in the Pharmaceutical Industry, and this webinar will be held by Dr. Lieven Kempenaers. He is the product manager for XRF. Lieven Kempenaers, he started his PhD in chemistry at the University of Antwerp in Belgium in 1999 with a focus on the use of XRF, X-rays in elemental analysis. He wrote many articles, of which one was awarded as the best article of the year 2000. In 2003, Lieven completed his PhD in chemistry and he used his XRF knowledge and the experience as an XRF application specialist and later on as a product manager for Pharmaceutical. After 5 years, he was relocated to the Asia-Pacific regional headquarters in Singapore and he took on the assignment as Asia-Pacific Regional Product Manager XRF. Back in the Netherlands in 2012, he used his Asia-Pacific experience as the Global Product Manager for the benchtop XRF instruments. And before I will pass control and the word to Lieven, I would like to inform you that this is an interactive program and that we encourage you to ask questions. You can raise your questions during the talk just by typing them in. And for this, you need to click on the 3 dots at the bottom right-hand corner of your screen. Just type them in and send them to the pane lists. And so Lieven will see the questions and he will get the answer to you. If we run out of time, then of course your questions will be answered later on by himself or by another specialist of Lieven Kempenaers. And this webinar will be recorded and the recording will be published to the website and we will then send you the link to this recording within the next few days. Another feature of the interactive program is that we have a poll, and I will open the poll right now. And this is for us very interesting to understand what the audience are in our webinars. And so I open the poll and we are very pleased if you could answer the questions. This poll will be open during the complete webinar. Yeah, so far for the introverts, and now I will pass control to Lieven to start your presentation.
Okay, thank you, Birgit. Can you hear me?
I can hear you very well.
Okay, good.
Yes.
Thanks. Well, thank you for the nice introduction and also hosting this WebEx. It's a great pleasure for me to say something today about X-ray fluorescence spectroscopy, called XRF, for the pharmaceutical industry and mainly for specific pharmaceutical applications. Let's see. Let's move. Yeah. So the content of this webinar will be a few topics. The first one, I will talk a little bit about some of the regulations and also some of the changes in the last few years. The second one is I will explain a little bit about XRF and the method itself as an alternative elemental method. The third topic for today will be a few slides on the total solution and some of the results that you can expect with the total solution. What can we offer? And the fourth topic is a case study done by Novartis and also us to see what can this solution offer for the pharmaceutical industry? And then, of course, the last few slides are a few conclusions to draw from this webinar. And then, of course, after that, we have time for question and answers. Okay, so let's first look at some of the regulations. When I start at the ICH, they describe the elements that need to be evaluated in a certain risk assessment that you do. And on the right-hand side, you see the column of all the elements in a certain class. There are 4 classes, class 1, 2A, 2B, and then also class 3. And the first 7 elements, they are called the big 7 because they are the most important elements. Also because the first 4 are from class 1, which are most toxic. And yeah, of course, all those 7 elements, they should be included in the risk assessment. Then, so from the class 1, we have the 4 toxic elements, which can be present as in the excipients. The class 2 are the toxic, or the class 1 is the highly toxic, and then class 2 is still toxic. Class 2A are the elements, cobalt, nickel, and vanadium. Those are elements that can enter into the end product because of some containment or containers or equipment, some wear elements, you could say. And class 2B elements, they have a lower probability that they will occur. That is also the list, and mainly those are the elements as the catalyst that can be used. And therefore, those elements are intentionally added. The other elements, for sure, are not intentionally added, but still have to be checked. And then you also have the class 3, the low toxic elements. So that's the list of the elements from the ICH. And so it's not only a list of elements, but the ICH also has the permissible limits for each element, depending also on the dose, daily dose that is consumed by the user, the customer. And so if you have a 1 gram daily dose, then the values that you see over here in the table can also be translated to microgram per gram, which is also the ppm levels. So those are the maximum limits, the maximum concentration in the end product. Now you have the US Pharmacopoeia, also the European Pharmacopoeia, but there were some changes since 2018 for the heavy metals screening quantification. So the US Pharmacopoeia chapter 232 states the limits of the elemental impurities in the drug product, and also the specifications, and they are in line, of course, with the ICH limitations. The chapter 233 also describes the elemental impurities, but then how to analyze and also the whole procedure. And there are 2 analytical procedures or methods that are included or specified as ICP-OES and ICP-MS. And also at the end of the chapter, it also states that alternative methods are also possible. And one of them is the XRF spectroscopy. Then there's also a new chapter in the Pharmacopoeia, which is chapter number 735. And that one is specifically on XRF spectroscopy. what it can do, what are the advantages, what are the limitations, and also specifically for the metal impurities testing. And that chapter describes the methods, the instruments that can be used for those testing with both wavelength dispersive XRF and also the energy dispersive XRF. So the two methods within the XRF technology. So those are the three, let's say, new chapters since 2018 for elemental impurities. Yes, so ICP is a very common method. And with this presentation, I am not going to convince you, and I'm also not telling you to stop using ICP, because ICP has many advantages, especially the very high sensitivity. But there are also some challenges that most of us know. And I just want to point them out here to make a comparison to the XRF technique. The first one is time from taking the sample to the end result can take hours, days, in some cases even weeks, because of the limited extra sample preparation. That's the determining factor. And that's also because in some cases the drug has to digest in the liquid, and in some cases it's very, very difficult, or the concentrations are very high. So then the digestion takes days, weeks in some cases for specific samples, and that means that the method, the whole method itself, will take some time. Also, daily recalibration for the elements of interest is also a must for ICP. And the method itself is also labor intensive because of the sample preparation. Also, the availability of staff that is properly trained to operate an ICP. That can also be a limiting factor to find the right people with the right knowledge, the higher education to operate the ICP and make sure that the right numbers are obtained. Also, the total, the capital cost of the instrument, including the sample preparation equipment, can also be a big investment. And also the availability of the infrastructure and also the utilities to operate an ICP, also with the gases, the fume hood, is also a necessary step for the investment. And also the availability of the operator. Once the operator is working on an ICP, he cannot do anything else, or working on the sample preparation, he cannot do anything else. So he's fully booked, one operator for one ICP on average. I mentioned the US Pharmacopoeia, the chapter 735, that also mentions alternative methods can also be used for chapter 232 and 233 for the metals testing. And one of them is the XRF spectroscopy. And I just want to point it out as it also has some nice advantages to make your life easier. So this is the chapter 735. It talks about the general application, the technique, and also the ability to do qualitative and quantitative analysis of those heavy metals in liquids, in powders, solid materials, slurries, those kind of things. So that's the chapter 735. And in one slide, in this slide, I want to explain what XRF, how the technique works. So the sample that you introduce to the spectrometer is bombarded with X-rays, with X-ray photon with a certain energy. And those are excited with an X-ray tube, with an X-ray source. A tube in the safe cases, the tube that you can switch on and off. Those photons that are generated by the tube, they bombard the sample, and inside the sample, depending on the elements in the sample, certain characteristic fluorescent X-ray photons are generated by the sample and emitted, and the detector collects those photons, finds out what is the energy of those photons and sort them out in a spectrum. And depending on the energy, you have the element, and depending on the height of the peak, you have the concentration. And of course, all of that is automatically done in the software. So so depending on the atoms in the sample, you get very specific photons with a very specific energy, and those can be sorted out through energy. What does a photon come from? Lead, or does it come from copper or arsenic or palladium, whatever. So that's in one slide how the XRF method works. Where would XRF work specifically in the pharmaceutical industry? Because XRF is used in many industries for many years. The food industry, but also the cosmetics industry, but also the petrochemicals industry, building materials industry, mining industries, even for 40, 50 years. People are using it, but in the pharmaceutical industry, it's rather new. And that's why I want to tell you about this method. So specifically in the pharmaceutical industry, where could you use XRF, or where does it make sense to use XRF? For sure, for elemental analysis, for quality control, for incoming goods, raw materials, and also quality control of the end product, the tablet, the drugs, the medicines. That can be used also for process control, checking certain elements that determine the process, but also checking counterfeit drugs. That's also possible in the R&D facilities or the main lab. Also screening unknown materials, samples that coming from the production site. That also happens sometimes, little particles, people don't know what it is. They bring it to the central lab. Can you tell me what it is? Where does it come from? XRF can also do a screening of those samples. And XRF can also be used as a pre-screening tool before the sample goes to the ICP for checking the elements, but mainly for checking the concentrations of certain elements so that you know what the dilution should be in order to protect your ICP method. So those are ideas for where does it fit in the pharmaceutical industry, in the process, R&D, quality control. So the XRF, what I mentioned before, in the XRF spectroscopy, there are two main technologies, you can say. The first one on the left-hand side is, we call it wavelength dispersive XRF, and on the right-hand side, it's the energy dispersive. I don't want to go into the detail how the method works, how the technique works, but I want to highlight, let's say the pros and the cons of the method. For on the left-hand side, the wavelength dispersive XRF is a very mature technology. It's there for at least 50, 60 years. It produces high-resolution data, also very low detection limits, also for the light elements, for the heavy elements. It's also a fast method. That's why it's also used in the metal industry, petrochemicals industry. One of the disadvantages is that because it's so fast, it also requires a high power excitation and high power source, X-ray tube. And that means with a high power, you excite the sample, but you also heat up the sample. And your case, in this case for the pharmaceutical industry, when we're talking about the active pharmaceutical ingredient, the APIs, those samples can be a little bit vulnerable. You want to keep it, you don't want to destroy it, well, you want to analyze it and continue in the process, in the R&D. And then wavelength dispersive, because of its high power, it will burn your sample, and it's not the best sample or the best method. Of course, if you're only checking raw materials and you have tons of the raw material, then it doesn't matter. On the right-hand side, you have the energy dispersive XRF. There, you could already see it in the picture. The method is simpler, and therefore it can also be made in a more compact way. So it also fits in a box that can be placed on a table. The wavelength dispersive are floor standing systems mainly, and because of the power, because of the cooling. On the right hand side, those methods are wavelength benchtop systems, and they also have low power. Everything is very compact. You don't lose a lot of photons when the design is well figured out. So that means if the instrument is compact, it can also be placed at line, like in the factory. It doesn't have to be in the central lab. So there already process control can happen. And because of the low power, it's considered as a non-destructive method. So the sample that you analyze this exact same sample is not destroyed, it's not heated up, can be taken to another method. It can also be ICP for further investigation, but can also be some completely other analytical method. So that's also an advantage. Yes, and those are the pictures that on the left-hand side you have the floor standing system, and the right-hand side you have the benchtop, more compact system. So going back to the list of challenges for ICP, this is exact same list, but here in the slide, for each topic, for each sentence, I put down a advantage of the XRF method compared to the ICP. So the first one was that the time from taking the sample to the end result can take a long time. Here in this case for XRF, the method itself, sample preparation and also the measurement takes 1 hour, 1.5 hours max. That's the total solution for all the elements of interest. The second one is with XRF, you can measure your powder, your API, your slurry directly. You don't have to digest it and put it in a liquid. That's not necessary. So that means that it's also much faster. For XRF is a very stable method. It's because we don't use gases or liquids. So that means that the instrument is stable over many months, years. Also the applications, once you calibrate the instrument, the XRF instrument also has to be calibrated for the elements of interest. Once you do that at the installation, you can run the instrument for months, for years, without having to recalibrate the whole instrument and use those standards again. Yes, so compared to ICP, XRF method is much faster. Also for the operator, it's also easier to use. So that means you don't have to have highly skilled operators specifically for this instrument. That's not necessary. It's also a very cost-effective method because of the benchtop and the sample preparation equipment, if necessary, is the only thing that is necessary. You don't have all the infrastructure, you don't need fume hood and expensive acids and liquids. And also, once the instrument is being used and measured, the operator can also do other things. The method itself is easy. The infrastructure is also very limited, and that means the installation of the instrument is also very basic. You don't need fume hood, you don't need water, compressed air, no liquid nitrogen is needed. Not even for this instrument, not even a vacuum pump is needed. Just a standard power cable. That's it, like you have with your computer, same electricity. So no complex at all. So this is, with this slide, I just want to draw the attention to the picture. So it's a benchtop system. It is compact. You can place it on the table, and it also has the sample changer for different samples that you can measure, different methods. It's also possible to mix those. And the instrument also, during the measurement, also spins the sample, the powder, to obtain a more homogeneous measurement of the whole sample. So how come in the past, the floor standing systems, they had a good performance, and at that time, 10 years ago, 20 years ago, the performance of the benchtop systems was lacking. But now we can say benchtop epsilon 4 in this case meets or achieves those requirements from ICH because of the new R&D activities that are ongoing from the tube, from the detectors, from the electronics. All of that makes the instrument more advanced. And you can see over the years, the instruments that we produce, the performance is increasing rapidly. So now we are at the point that even a benchtop system can reach those very low limits, a few ppm of quantification limits for all those elements. I talked about the instrument, but sorry, I have to make sure I don't run out of the battery. Yes. I talked about the instrument, the epsilon 4. I also want to say something about the whole method that we offer, and then also some results, what we achieved. We have also the standards. I mentioned we have to calibrate the instrument, and that's also what we did in the factory. We calibrated the epsilon 4 for those 17 elements that are present in those setup samples that we make ourselves. The basis is cellulose. But also other matrices bases are also possible. There are also different ways of sample preparation. As I mentioned, it's very simple. On the left-hand side, you have for the low amount of sample in the milligrams, 100 milligrams, 200 milligrams. There we have a special disposable cup where you can put the powder and measure directly. When you have a few more, when you have more amount of sample for the analysis, then you can also use a disposable cup where with a spoon you can also place 5 grams in the cup and analyze directly in the instrument. Or if you want to obtain the best results, even with the powders, and you have a lot of material, you can also press the powders so that the surface is also more flat and that the method is more repeatable. So it depends on your requirements, what is necessary. And here are the different steps from the left to prepare your samples. This is how to measure, and this is the procedure, how to measure the sample. You prepare the sample in any way. You load it in the instrument. You select the application that is there. The instrument is calibrated. You type in the name of your sample. You push the green button, measure. Then, of course, you have to select the position. Where did you put it on the sample changer? And then after 5 minutes, 10 minutes, half an hour, you obtain the results of those elements of interest. In this case, those are the big 7 elements. So this is just a flow of what the procedure is for the measurement. Also, yes, I mentioned, let me show you some results. What can you expect? What we did is, what we did here is the setup of the all those 17 elements with the different suitcases, with the standards, and the total measurement time was 1 hour. And then we reported the limit of detection. So the limit of quantification, that's a better way. You have to quantify it. So here, this is an overview of not all of the elements that we analyzed, but to give more of focus on the most important elements, and this is the toxic elements and also the catalyst residues. This column describes the limits set by ICH, and these are the limits when you convert it back to 2.5 grams of daily doses. So that means that you have to be able to quantify 2 ppm in your sample. Then of the total samples. Then what we did is we set up the method for pressed pellets, also for the loosed powders, just in the disposable cup, and then also with the small mass, the 100, 200 milligram. In this case, it was 100 milligrams that we used. And you can see, that's why all the numbers are in green. Looking at the ICH limits, we pass all the elements for the limits. And the other elements are even easier because they are less toxic, so those levels are even higher, so easier to achieve. The most critical ones are the first ones, the big 4 of the most toxic elements, and we achieve all those within 1 hour of measurement, including the sample preparation. So those are nice results. Also looking at the accuracy of the method, so validating the method with a sample where we know the concentration and we analyze it as an unknown sample and compare it against the certificate, where you can see around the 20 ppm or 50 or 100 ppm was in the sample. And then we also achieved 2018, 2020, and then 51 ppm against those elements. So nice accuracy for all those elements. And you can also make it in a visual way, where you calculate the recovery rate in percent, where everything is well within the limits between the 50 and the 150%. Even within the 80 and the 120%, it's well within those limits of recovery for all those elements that we investigated. So that's the accuracy. Then also looking at the accuracy for just 100 milligrams in this example. And again, where we have the given concentration, what it should be, and also the obtained concentration when we measure the sample as an unknown sample and compare the results. So those are measurements that we did ourselves in the factory with the standards. I want to point out to a case study that was initiated by Novartis using our instrument. They did not use our standards, they used our instrument. They had their own way of setting up the whole method. I'm not going into all the details of the article, but just mentioning that just a few things I want to point out. So the paper describes, first of all, the motivation. Why would they look at XRF in the risk strategy in Novartis? And also what they did is they found or developed a method by testing a subset of a big group of 400 solid dose products. And so they also had their target at 2.5 gram daily doses. That's all written in the paper. The interesting part is that the authors, they also look at other available methods, and they conclude that they chose the benchtop ED XRF instrument over ICP-OES, even the laser ablation ICP-MS, handheld XRF, and also WD XRF as alternative methods, but they chose the benchtop ED XRF method as the method that suits for them. In the article, they also describe why not the others. They mention not ICP-MS or ICP-OES because it's a complex method, time-consuming, and high running costs. And also they mention specifically, and that was also the study, if there is silicon oxide or titanium oxide from the coating of the tablet, if that is present for ICP, then you have to use toxic fluoric acid to dissolve everything into the liquid. And they thought that as a big disadvantage. They also looked into the laser ablation, where they said that the amount of mass that is analyzed is too small to get a homogeneous representation of the final product. They also looked at the performance of the handheld XRF, and they said that's not good enough because the sensitivity is not good enough to achieve those low ICH detection levels. They also looked at WD XRF, and they described that, yes, it's also a powerful technique, but yes, the high power will make the method a little bit more complex when certain elements like mercury, they disappear or they evaporate because they are more volatile. And also they mentioned that the WD XRF method only does looking at the peaks, the height of the peaks, and doesn't do full scanning for troubleshooting later on. They also mentioned the advantages of ED XRF as it's a simple method, and in this case for them specifically, they said it's easy method for the sample preparation because we only have to crush the end product, the tablet, and press it into a pellet. And also that means that they analyze the full tablet. That's also what the customer is using, and taking the full method. And that means that includes the API, the excipient, and also the coating. And sometimes the coating can be titanium oxide. And that makes things complicated. But they found a nice, simple way also with XRF, and also the very low operating costs. And here in this picture, this is what they mentioned, that I made a drawing myself to make a more schematic representation of in the spectrum. They are interested in the vanadium because vanadium is one of the elements of interest. The titanium as such is not according to ICH. It's not of interest, but vanadium is. And then they noticed that when you're analyzing a sample with very high titanium oxide in it, you obtain a very high titanium K alpha peak in the spectrum, and also titanium K beta peak. They always come together in a certain ratio. Then the authors, they say, when you're interested in low concentrations of vanadium, you will have a problem. But of course, if there is no titanium in the sample, then you can just use the vanadium K alpha peak to determine the concentration of vanadium in the end product. If there is titanium at a high concentration in the sample, then you can switch over to another calibration that is focusing on the vanadium K beta peak, which is freestanding and doesn't give any errors. And so they determine the limitation of if the titanium is above 1000 ppm, then it's better to switch over. And within the epsilon Epsilon 4 in the Epsilon software, there is the feature, the automatic program selection, that automatically looks first a few seconds, looks at the titanium peak, and then determines and selects the best method to use for that kind of sample. So they used their own standards. They spiked those big 7 elements in low concentrations between 1 and 20 ppm. And then the total method per sample was half an hour. Okay. These are some results that the people in Novartis obtained with the instrument and also with their method. And I just want to point out that for the big 7 elements, within the 30 minutes, they achieve all the values, or at least the limits of quantification, except for cadmium, which is on the edge, and that can also be changed by increasing the measurement time to 45 minutes, and then it's already solved. And this is the LOQ that they determined, also according to the ICH method with 10 times the relative standard deviation. And that's how they determined. Then in another way, they also determined the limit of quantification by looking into the software, into the instrument, what is the peak height, what is the background, and they also calculated in that way. And there's a nice correlation between the two, and that gives you an indication what are the limit of quantifications for those elements in half an hour with the method. And these are some pictures that are also shown in the article of those 7 elements on the left-hand side. This is the recovery rate, well within the limits, even at the level of 2 ppm, 2 microgram per gram. And on the right-hand side, it's the same for the other elements, the less critical elements, at the 10 ppm level. So this is for the recovery rate, and this is for the repeatability. So 10 measurements after each other, checking all those elements at the 2 ppm level and at the 10 ppm level, and it's all within the 15% relative standard deviation. So they concluded very nice results. The method is easy, is cost effective, so this is a nice fit for us in the company. That is about the more the details of the article. But that is was an evaluation of the method for Epsilon 4 for pharmaceutical samples. Just have a few last slides on what are the possibilities of a screening tool, also XRF, for catalyst and metals with Epsilon 1. So Epsilon 1 is, let's call it the smaller brother, the small brother of the Epsilon 4. And in this case, this is the Epsilon 1 with a built-in computer with a touchscreen. There's no sample changer. It's a one position for a few samples per day that you want to measure. In this case, specifically for the catalyst residues, and it takes 10 minutes to screen, to check the concentration of those 5 catalysts, one of the 5 that was used in the end product or intermediate API samples. And also here, the procedure is very simple. And also in this case, we use only 100 milligrams of sample, made a disposable cup and put it in the instrument. That's picture 3 over here in the instrument. And then with the touchscreen, type in the name of your sample and push the go button, measure button. And then in this case, after 10 minutes, you receive the elements and the concentration. This is an example of the results. So the calibration of palladium from 0 to 200 ppm. In this case, that's how we have set it up. And also the nice thing that I want to point out is the good comparison between the epsilon 1 data and the ICP-MS data. So we also sent those samples to a third party, analyzing the samples with ICP-MS. And so we obtained average concentration for a certain repeated measurement, also with the RMS value. So the average concentration and the standard deviation. And then at the bottom is the corresponding concentration for each element. And it gives a nice correlation, 50, 51. This is a specific sample. It's an artificial sample, spiked sample. But you can see that there's a nice correlation between the XRF method and the ICP method. Okay, and I just want to mention, talking about the total method of the XRF benchtop instrument for pharmaceutical analysis. We also have standards available to set up the calibrations, depending on the elements, depending on the matrix of your interest. And we also have, on the instrument, we have extra software, the enhanced data security software for the audit trailing, specifically for the FDA 21 CFR Part 11, that we are compliant to that method, so that everything is traceable. Results cannot be cannot be deleted, and if you want, then a copy is made. You know, you have to log in with a username and a password. You have different rights, those kind of things. So everything is traceable, and that software is available also on the benchtops. And also the installation qualification document and operational qualification documents are available if necessary. So that's, yeah, that would make it a complete package. Okay, so we reached the end of this webinar. I hope you picked up certain things, or it sparked some questions or nice ideas also in your company, in your process. So this summary slide talks about the advantages of the benchtop XRF over ICP and AAS. Of course, it's a repetition from this presentation. So first of all, the sample preparation is simple and fast, because you can measure the powder directly. You can also do a liquid, you can also do a slurry. That's one. Of course, with the fast sample preparation, that means that the whole method is fast, and that means also the operational cost of the method, including sample preparation, is also very low compared to ICP. And also the utilities are very limited. It's only electricity that is necessary. And also with the benchtop XRF method, you don't have to recalibrate the whole instrument in the morning before you can do anything, because that is monitored automatically. And everything is stored, what the instrument is doing. And as I mentioned, the benchtop XRF method can be considered as a non-destructive analytical technique, so that even after the measurements, you can take that sample for further analysis in ICP or somewhere else. And then my last slide as a summary for the, what is the value of the benchtop XRF? First of all is when the method is fast, that means that you can have frequent analysis in the process control, because you can also, it's a benchtop. It doesn't need any infrastructure. You can place it in the factory, close at the heat, let's say, where everything is happening. Also, the method itself is fast, so that means that the feedback loop is also very short. You don't have to wait for days or weeks before you get any result back. It's within 1 hour, 1.5 hours, you know your result. And XRF can reduce the workload of ICP. I'm not, with this presentation, I'm not saying to stop using ICP, not at all. I'm just saying that XRF can help you in certain operations and in this case also reduce the workload of your ICP, which is most of the time fully loaded with samples. And also for when using the benchtop XRF, it's also much safer for the operator because he doesn't have to use dangerous chemicals. And also the glassware that has to be used with ICP. Okay, so very good. This is my last slide.
Thank you. Thank you so much, Lieven. Thank you very much. This was a very interesting presentation, and we've got lots of questions. So therefore, we will start directly. I will check with in the question and answer session. So I see. What is the exact angle between the source incident beam and the detector? Looks like 45 degrees.
Yes, that's a very technical question. I don't want to go into all the details, but it's around the 45 degrees, which gives the best sensitivity according to the law of physics. Yeah.
In which stage of pharma industry is it required most? API, formulations, or final product?
Yeah, is it required most? The at the end, the final product has to be checked for all those 7 products. That's a must. And of course, when to make your life easy, I would say if you have to check your end product and you find something, it's already too late. It's good to do those double checks also during the process to make sure that not, let's say, contaminant excipients are introduced into the production. So the further you go upstream in the process, the better it would be. Of course, that's my opinion. Also to lower the cost when there is an issue at the final product and you have to stop the production. That's more costly.
Thank you. And the next question, what are the major challenges of XRF in the element of interest as far as pharma industry is concerned?
In the element of interest, the major challenges, one was the, as I explained, the challenge of analyzing the vanadium when in the presence of high titanium. But I mentioned that the software is clever enough to select the other peak in the spectrum. Other challenges could be that the light elements, but they are not of interest for ICH or also the US Pharmacopoeia, but sometimes also light elements are of interest. They can also be analyzed, but those concentrations are, you cannot achieve 1 or 2 ppm for the light elements, let's say magnesium. And that's more in the 50 ppm, 100 ppm levels. That's also possible. So that would also be a challenge towards the lighter elements.
Thank you. Do we have any set of standards for calibration or is it available in the market?
Yes, we produce those standard samples ourselves. So that means we are in control of the elements, the concentration range, and also the matrix. In this case, we started with the cellulose. But yes, we have those because the reason why we made it is because we saw in the market that they are not available. In the case study that I mentioned, Novartis made their own, let's say, in-house standards themselves.
Thank you. How to decide on VD or ED? Is it only on sample throughput or sensitivity and elemental concentration also matters?
Yeah, WD or ED. Yes, it depends on the elements of interest. If you're more focusing on the elements from ICH, which we consider more heavier elements, metals, then ED would be a better choice. And also what you want to do with the sample afterwards. Then with the ED, you don't burn your samples. Yeah, so specifically for the pharmaceutical industry, the ED method seems to be more suitable than WD method.
Thank you, Lieven. And what type and thickness of polymer films are optimum?
Yeah, the principle with XRF is the thicker the foil that you use for, I assume, your disposable cup, the more absorption you get. That means the less intensity, so less sensitivity for your elements. So we always advise a good, strong foil that is transparent for the X-ray photons, but still strong enough. Yeah, polymer films. Yeah, so sometimes a Mylar is available on the market. That's only a few microns thick. It is strong enough. Also, polypropylene is sometimes used, which is 6-micron films that can also be used.
Next question: what is meant by recovery rate?
Okay. Yes, that's a typical. How do you say that? That's a typical way of presenting results in the ICP world. And in here, we try to do the same. So that means that you have a sample with a certain element at a certain concentration. You measure that as an unknown sample, and that means that you obtain a different number, a different value. And the difference between the two is the recovery rate. So the closer you get to the real value, to the initial value that is in the sample, the closer you achieve the 100%. That's the goal. And of course, with ICP, the recovery rate is a difficult topic because you also have elements in your sample and you want to digest all of them. And that means that you might lose a lot of concentration, a lot of material during the sample preparation method. And XRF, the sample preparation method is much simpler. So that's why the recovery rate is automatically much higher. We don't lose material on the way towards the measurement.
Thank you, Lieven.
Yeah.
Can we measure mercury in the sample by ED?
Yes, I forgot to mention that. It was somewhere on the slides. It was also the conclusion of in the article from Novartis. They concluded, yes, we can measure the mercury in the sample. Mercury itself is an easy element for XRF, high sensitivity. The problem, what they found is that in one of their standards they used, the mercury became volatile and the concentration was much lower than expected. So they saw some error. So the mercury itself is volatile, but for the instrument, it's not a problem. And when talking about benchtop ED-XRF with low power, then the mercury doesn't evaporate during the measurement. That's also an advantage. So yes, you can measure mercury.
The next question, we are interested in monitoring trace elements in our incoming materials. Would ED-XRF be suitable for measuring levels of zinc, manganese, copper, iron in various raw materials? What would be the LOQs for these elements?
Let's see. I always say it depends, but of course that doesn't that doesn't work as an answer. Yes, so looking at those elements, the zinc, manganese, copper, iron, yes, those elements are, I would say, easy elements, especially for ED-XRF. That's in the sweet spot of the detector, so there's no issue there. What would be the limit of quantifications for those elements? That depends on the matrix. If we're talking about organic matrix, kind of cellulose, I would say the LOQ, that's also what I mentioned. I think that was for nickel. The LOQ was 1 or 2 ppm, and that is in 10 minutes, 30 minutes measurement time. The principle of XRF is the longer you measure, the better your results will be, the better detection limits as well. But of course, at a certain point, you cannot measure. If you measure for 1 hour or you measure for 1 day, the result will be exactly the same. But there is a difference if you measure for 2 minutes or 20 minutes. So yes, few ppm limit of quantification for those elements, yeah, in various raw materials. It's possible.
Thank you. Would it be a problem to measure an intact tablet with a curved surface without grinding or grinding? How deep is the penetration depth in general? Would one get more than just the coating of a tablet?
Again, that depends. The curvature itself is not an issue. Because yeah, we have customers who do that. They're interested specifically on the coating. Then they cut the tablet and they look also inside. That's also possible. They cut the tablet in half. XRF, yeah, radiation. It's dangerous because it penetrates through a lot of material. Yes, but to a certain extent. If the coating, so depends on the material of your coating. If the coating is titanium oxide, and also depends on the thickness of your coating, it will be difficult to also analyze elements of the excipients or the API behind the coating. If your coating is like magnesium oxide, so a much lighter element, then it would be easier for the elements to be measured behind the coating. Yeah, the easiest would be to double check. But I know customers who who slice the tablet, measure the coating, measure the inside just to make sure that they have the difference. And in Novartis, there was the different. There they grind the whole tablet, and then they have the whole result of all the elements of the whole tablet.
Thank you. Is XRF semi-quantitative compared to ICP?
No. It's both quantitative, like ICP. XRF can also be used as a semi-quantitative tool, and the screening tool. Which is more difficult with ICP because you have to know the elements and the concentrations up front. With XRF, that's not necessary. You can put any sample in there, and then you can use the screening method, which is, we call that semi-quantitative. And that's plus or minus a few elements, concentrations. But once you have your instrument set up properly with calibrations, then it's equally as quantitative as ICP. Because with ICP, you also need standards to calibrate the instrument.
Thank you. And I hope now I will pronounce it correctly. With XRF, is the LOQ for a given element impacted by the sample matrix, for example, in dextrose versus in sodium phosphate?
Yes. The yeah, sort of, yes. So the LOQ will be different when you have a different matrix, when the matrix is, let's say, yeah, like in this case, dextrose or sodium phosphate, it's slightly different. But still, we're talking about the LOQ of a few ppm difference. It's not that the LOQ is 2 ppm and with the other one it's 2%. That's not, but there will be a difference that the heavier the matrix, the higher the limit of quantification will be, because the matrix will also absorb the photons coming from the elements reaching the detector.
Thank you, Lieven. We have now 2 more questions in the question answer section and 4 more in the chat. So, but I think we want to answer them all. Yes, right now we have 5 minutes over time, but so many participants are still joining, so we get your answer right now. Is gain control necessary to perform periodically?
Yes. It's always good to periodically, once every month, to just do a check with a sample, validate the calibration. Also, you want to know for yourself if the calibration is still alive. So yeah, in a periodic way, I would do that once a month is maybe good enough, just to double check if the calibrations are still alive and valid.
Okay, next question. What does tube flash indicate?
Oh, that's a very technical question. The tube flash means that that means that that also has something to do with the generator, with the cable, and that means that sometimes when generating X-rays using very high volts, kilovolts, that sometimes the tube flashes, and yeah, for what is it, a millisecond, where the tube doesn't produce any photons. And in some cases, it's fine. When you see that in older tubes, that means that the tube is ready for replacement. That can also be an indication when it happens more and more.
Thank you. Next question. Is there a performance parameter to evaluate the instrument sensitivity?
I don't. Is there a performance parameter to evaluate the instrument sensitivity? I don't understand the question. Performance parameter. Yeah, how do you evaluate the instrument sensitivity? I think it should be the other way around. What is what do you need? What are the elements of interest? What is the sample throughput that you want? And then we as a specialist, we can give an advice of, yeah, this is the best instrument, Epson 1, Epson 4. In some cases also the floor standing system with the high power, also for the high sample throughput. That can also be possible, but it should be the other way around. So how do you evaluate it? It's more, is that specific instrument sufficient for solving your problem?
Thank you. And now I will go to the chat section. Okay, I start. Can you please explain the safety issues related to X-ray risk to people around the machine?
Okay. Well, actually, there are no safety issues. So X-rays are dangerous only when they are not contained. All the instruments that we make within the company, they're all certified against the most stringent norms, German norms, Australian, Canada norms. So it should not be higher than, what is it, 1 microsievert per hour in a certain distance. I've been using the company's instruments for 20 years. I have 4 children, so but yes, we in the factory for each instrument that we produce, we double check with the counters if there is any leakage after the manufacturing, and then we ship the instruments to the customer, and there our service engineer does the same check again before you can operate it. Then we double check if everything works fine, if there are no X-ray leakages. So that's all taken care of. Yeah, we take that very seriously, especially for operators who use it on a daily basis.
Thank you, Lieven. Which is the minimum LOD achievable with the lowest sample quantity?
Again, that depends. But so I showed you some results when using 100 milligrams of sample. And then we were interested at, I think in the case was 17 elements within 1 hour. If you say, I want to, I only have 50 milligrams or 20 milligrams of 10 milligrams, it's still possible, but the principle of XRF is the more material that you have, the better response you will get from your instrument. The more photons you generate, and then the more photons the detector will see. So that means the higher the peaks. If you only have 20 or 50 milligrams of sample, you can compensate by measuring longer, where you achieve similar results. But of course, there is a limitation. We checked up to 20, 30 milligrams. It was still possible, but of course, then the limit of quantification also goes up. And then you cannot expect to see less than 1 ppm of an element when the material is so low.
Oh, there are coming more and more questions. For quantitative analysis, we need to use specific amount of powder or pellet of specific thickness, right?
Yes, for the sample preparation, it is when you use the same sample preparation as for your standards, you will obtain the best results. So that's also part of your standard operating procedure. Every day, when you want to press your sample, it should always be the same amount, let's say 10 grams. That's also easier for the operator. If it's always 10 grams, it always has the same height. It will give the best results, most reproducible, yes.
Can it replace atomic absorption spectroscopy?
Yes. Again, it depends on the concentration limits. Atomic absorption is very specific for certain elements also in the lower concentration. So below the ppm, so in the ppb range. XRF is limited up to 1 ppm or half a ppm, and then XRF starts to struggle. So you could replace the AAS method when the concentrations are above the ppm level.
Thank you. And now I think it's more a comment than a question. It's better to use WD XRF than ED XRF for more sensitivity for some element.
Yes, for the light elements. So if we're talking about magnesium, aluminium, silicon, sodium, then a WD-XRF is a better method to use than ED-XRF. Yes, that's correct.
And therefore, the same person, therefore, it is more convenient to use WD-XRF in pharmaceutical industry to have more sensitivity, correct?
That depends on the application, because as I mentioned before, with a WD-XRF, the power is always much higher than with ED-XRF. So that means you heat up your sample, you burn your sample with X-rays, the matrix starts, the crystal structure starts to change. We also double check that with XRD spectra. So WD and XRF is not always the best choice for the pharmaceutical industry, especially when you have the heavy elements. The sensitivity of the heavy elements is better with ED-XRF. Yeah.
Thank you, Lieven. Thank you for
You're welcome.
answering all the questions. And thank you very much to all participants for this great audience. Thank you for interacting so much with our specialist, and I really like it. I thank you very much. So if you still have questions, please send them to events@malvernpanalytical.com and we will forward them correspondingly. So this was a great webinar, and I wish you a very nice day, a nice evening for us here in Germany and the Netherlands. And I hope to meet you again in the next webinar. And yes, I hope you enjoyed it, and thank you very much. And one comment, thank you very much for answering our poll questions. And there will be a very short survey when you leave the webinar, and this will help us improve our webinar program as well, if you could take some moment to answer these. So
yes, thank you very much, Birgit, for hosting. You did a very good job for hosting this webinar. It was a pleasure working with you, and see you next time. I'll talk to you next time.
Thank you very much, Lieven. It was a pleasure working with you, being the host for today. Thank you to all, and bye-bye.
Bye-bye. Thank you. Bye-bye.